Welcome to G Fun Facts Online. The history of life on Earth is written in the language of DNA, a dynamic, ever-changing script of adaptation, resilience, and survival. Yet, for many species, this genomic library is being rapidly burned. When a species experiences a catastrophic drop in population size due to habitat destruction, disease, or extreme weather events, it undergoes what biologists call a population bottleneck. The surviving individuals represent only a tiny fraction of the original genetic diversity. In the aftermath, the population faces a peril almost as deadly as the initial disaster, the extinction vortex. In Australia, a continent home to the greatest diversity of marsupial fauna on the planet, the stakes are exceptionally high. The region also bears the grim title of having the highest mammal extinction rate in modern history, with dozens of unique species vanishing since European colonization. For marsupials, from the iconic koala and the ferocious Tasmanian devil to the diminutive mountain pygmy possum, the threats of habitat fragmentation, invasive predators, and rapid climate change are unrelenting. However, a scientific revolution is fundamentally changing the landscape of conservation. By merging traditional wildlife ecology with advanced molecular biology, scientists are pioneering a new frontier known as genomic rescue. Through whole genome sequencing, targeted genetic translocations, biobanking, and cutting-edge assisted reproductive technologies, conservationists are no longer just documenting the decline of these unique mammals. They are actively engineering their survival. To understand the sheer power of genomic rescue, one must first deeply understand the invisible genetic threats that plague small populations. A population bottleneck drastically reduces the number of breeding individuals, effectively acting as a sieve that filters out vast amounts of genetic variation. Genetic diversity is the raw material of evolution. It provides a population with a diverse toolkit of gene variants, known as alleles, that can offer resistance to novel pathogens, adaptability to shifting temperatures, and resilience against environmental stressors. When a bottleneck occurs, this genetic toolkit is stripped down. The immediate consequence is a massive increase in genetic drift, which is the random fluctuation of gene frequencies. Genetic drift can cause highly beneficial alleles to vanish entirely by pure chance. Furthermore, as the population shrinks, related individuals are inevitably forced to breed. Inbreeding significantly increases the likelihood of homozygosity, a state where an offspring inherits two identical copies of a specific gene. If these copies happen to be deleterious or harmful, recessive mutations, the resulting animal may suffer from compromised immune function, severely lower fertility, and profound developmental defects. This phenomenon is known as inbreeding depression, and it initiates a devastating positive feedback loop. The population shrinks further due to poor overall health, which inevitably leads to even more inbreeding, which then drives the population down yet again. This inescapable downward spiral is the true definition of the extinction vortex. For decades, conventional conservation wisdom held a deeply pessimistic view. Once a population fell below a certain critical threshold and lost its foundational genetic diversity, it had effectively reached an evolutionary dead end. Genomic rescue, however, flips this paradigm entirely on its head, proving that strategic human intervention and natural demographic rebounds can actually restore lost genetic vitality. The absolute proof of concept for genetic rescue in marsupials was vividly demonstrated in the high alpine ranges of Australia, starring one of the world's rarest creatures, the mountain pygmy possum. Restricted to isolated freezing boulder fields, these mouse-sized marsupials are highly vulnerable to habitat degradation and feral predators like foxes and cats. By the year 2005, one specific population at Mount Buller in Victoria had experienced a devastating collapse, dwindling to fewer than 20 known individuals. Because of geographic fragmentation, this specific population had been completely isolated from other mountain pygmy possums for roughly 20,000 years. Their gene pool had reached a disastrously low ebb, and extinction seemed not just likely, but imminent. In a bold conservation experiment, a team of researchers led by Dr. Andrew Weeks from the University of Melbourne decided to implement a targeted genetic rescue strategy. In 2011, they captured 6 healthy, genetically diverse males from a much larger population at Mount Hotham and introduced them directly to the dwindling Mount Buller group. Historically, conservationists have been incredibly hesitant to mix long isolated populations. This hesitation stems from a theoretical risk known as outbreeding depression. a scenario where hybrid offspring might lose highly localized adaptations or suffer from complex genetic incompatibilities. However, the mountain pygmy possum rescue completely shattered these historical fears. The introduction of fresh DNA sparked an immediate and profound biological revitalization. The results were nothing short of astonishing. The first-generation hybrid possums displayed remarkable hybrid vigor, a biological phenomenon also known as heterosis. They grew physically larger, the female hybrids produced significantly more pouch young, and they enjoyed much longer lifespans compared to the non-hybrid original residents. Driven by this vital influx of genetic diversity, combined with aggressive predator control and dedicated habitat restoration, the Mount Buller population experienced rapid demographic growth. By recent estimates, the population had swelled to over 200 adults, making it the largest ever recorded at that specific site, and it boasted the highest levels of genetic diversity in its modern history. The Mountain Pygmy Possum Project served as a global beacon, proving definitively that targeted gene flow could rapidly pull an endangered marsupial out of the extinction vortex. While the mountain pygmy possum faced isolation and invasive predators, another iconic marsupial, the Tasmanian devil, has been fighting a highly visible, gruesome battle against a totally unique biological anomaly, devil facial tumor disease. Discovered in the mid-1990s, this is a contagious cancer that spreads directly through the biting behavior typical of devil social interactions. The disease has decimated wild populations, causing catastrophic declines of over 80% across the entire island of Tasmania. Early genomic analyses suggested an incredibly bleak future. Researchers discovered that Tasmanian devils had suffered prehistoric population bottlenecks thousands of years ago, during periods of extreme climatic instability. This ancient history left them with historically low baseline genetic diversity. This glaring lack of diversity was heavily implicated in their massive susceptibility to the contagious cancer. Because the devil's genomes were all so similar, their immune systems completely failed to recognize the infectious tumor cells as foreign invaders. Fearing the total and irreversible loss of the species in the wild, the Save the Tasmanian Devil program rapidly established what they called an insurance metapopulation. This was a massive, highly coordinated captive breeding network spread across dozens of zoos and protected sanctuaries, such as Maria Island. The primary goal was to preserve the remaining genetic diversity in a strictly disease-free environment for future rewilding efforts. Recently, incredible breakthroughs in genomic sequencing have painted a much more optimistic picture of the devil's long-term future. An extensive 2022 study published in the prestigious journal iScience, led by Dr. Catherine Farquharson and Dr. Carolyn Hogg, meticulously compared the full genomes of 830 wild devils and 553 insurance population devils. The researchers analyzed over 500 critically important functional genes, specifically those intimately linked to reproduction and immune response. The findings of this massive genetic audit were a monumental victory for conservation management. The study proved that the insurance population successfully captured and perfectly maintained the genetic diversity of the wild populations. Lingering concerns that captive devils were becoming genetically naive or losing crucial wild alleles over time were proven largely unfounded. Furthermore, and perhaps even more amazingly, scientists have identified clear footprints of natural selection in the DNA of the surviving wild devils. The wild populations are rapidly evolving in real time, showing distinct genetic shifts in loci directly related to immune response and cancer resistance. Armed with this high-resolution genomic data, managers can now continuously optimize translocations, carefully supplementing wild populations with captive-bred individuals to maximize genetic variation and actively bolster the species' evolutionary arms race against the tumor. Now this brings us to perhaps the most globally recognized Australian marsupial of all, and one that has suffered a truly tragic recent history, the koala. Once incredibly abundant across the continent's eastern seaboard, koalas have endured centuries of intensive hunting for the fur trade, devastating and widespread habitat clearance, and rampant, debilitating diseases like chlamydia and the koala retrovirus. To make matters vastly worse, the catastrophic Black Summer bushfires of 2019 and 2020 further decimated their remaining numbers, incinerating vast tracts of vital eucalyptus habitat and pushing several regional populations to the absolute brink. Due to historic, human-induced population crashes, many koala populations, particularly those in southern Australia and Victoria, experienced severe genetic bottlenecks. For example, extensive modeling indicates that the Victorian koala population once crashed to a mere 102 individuals before eventually expanding to larger numbers in subsequent generations. According to classical population genetics, such an extreme, narrow bottleneck should have left these southern koalas genetically impoverished, completely riddled with deleterious mutations, and highly vulnerable to extinction. However, a groundbreaking whole genome study published in the prestigious journal Science in March 2026 has radically reshaped our fundamental understanding of genetic risk and evolutionary resilience. Led by Dr. Colin Ahrens, a dedicated team of researchers analyzed complex whole genome sequencing data from 418 koalas representing 27 distinct populations across Australia. Their findings completely challenged long-held conservation assumptions by demonstrating that genetic recovery from severe bottlenecks is not only theoretically possible, but is actively occurring right now. The researchers discovered that rapid demographic growth, meaning a massive population surge following a devastating crash, can actually act as a powerful evolutionary reset button. As the bottlenecked Victorian koala populations began to expand, the fundamental mathematical realities of their genetics shifted. The key underlying mechanism driving this incredible recovery was recombination, the natural biological process that randomly reshuffles DNA during the formation of sperm and egg cells. In small, stagnant populations, deleterious or bad alleles are often physically linked to beneficial ones on a chromosome, acting like an anchor and dragging the overall fitness of the entire population down. But as the koala populations grew rapidly in overall size, increased rates of recombination physically broke apart these bad allele combinations. This continuous genetic reshuffling, combined with the gradual, natural introduction of new mutations over time, successfully diluted the harmful genetic load and allowed rare, functional alleles to reappear and spread throughout the population. This landmark 2026 discovery provides an extraordinary and much-needed message of conservation optimism. It proves definitively that a bottleneck does not guarantee a slow, inevitable march to extinction. If conservationists can mitigate external, human-driven threats, such as actively protecting remaining habitats from bulldozers and bushfires, and effectively managing disease, and then allow the population size to rapidly expand, natural evolutionary processes can actually restore the long-term genetic health and adaptive capacity of the species. We just have to give them the physical space and time to bounce back. But while whole genome sequencing allows us to meticulously read the genetic code of endangered species, we still desperately need physical, practical mechanisms to propagate this vital diversity. This is exactly where assisted reproductive technologies become absolutely essential. Assisted reproductive technologies encompass complex clinical techniques like artificial insemination, in vitro fertilization, and sperm and egg cryopreservation, often referred to as biobanking. While these methods are totally routine in human medicine and modern livestock agriculture, they have historically been incredibly difficult, almost impossible, to adapt to marsupials. Marsupial reproduction diverges radically from placental mammals. They give birth to highly altricial, meaning severely undeveloped, young after incredibly short gestation periods, often just a matter of a few weeks. The vast majority of their early development occurs completely externally in the mother's pouch. Because of their highly unique hormonal cycles, specialized reproductive tract anatomy, and completely different embryonic development, traditional mammalian-assisted reproduction protocols simply do not work on marsupials. But a colossal leap forward was achieved in February 2025 by a pioneering University of Queensland team led by Dr. Andres Gambini. His team successfully produced the world's first kangaroo embryos using in vitro fertilization and intracytoplasmic sperm injection, a process where a single, selected sperm is injected directly into a mature egg under a microscope. Because eastern gray kangaroos are relatively abundant, they serve as a perfect, low-risk proxy model to refine these incredibly complex techniques without risking the lives of critically endangered animals. Dr. Gambini's spectacular breakthrough provides a critical, tangible blueprint for the future of marsupial conservation. By successfully demonstrating that marsupial eggs and sperm can be safely collected, cultured, and fertilized in a controlled laboratory setting, researchers are laying the crucial groundwork for establishing frozen zoos. In the near future, conservationists will be able to freeze the gametes or even skin cells of genetically valuable, critically endangered marsupials, such as the northern hairy-nosed wombat or the leadbeater's possum. If a wild population suffers a massive, sudden loss of diversity due to a localized bushfire, scientists could thaw these safely preserved cells, create living embryos via IVF, and use surrogate mothers of a more common species to rapidly inject that lost genetic diversity straight back into the wild population. The ultimate frontier of this genomic rescue, however, is synthetic biology and the controversial but incredible science of de-extinction. While the idea of bringing back extinct species definitely captures the public's imagination, the underlying technologies being rapidly developed in these pursuits have immediate, life-saving applications for living endangered marsupials. This incredible synergy is perfectly encapsulated by the massive efforts of Colossal Biosciences, a cutting-edge company actively working to resurrect the thylacine, also known as the Tasmanian tiger, the apex marsupial predator that was tragically hunted to extinction by 1936. In late 2024, Colossal, working closely alongside Dr. Andrew Pask from the University of Melbourne, announced they had successfully assembled a near-complete, chromosome-level thylacine genome using degraded DNA extracted from a 110-year-old preserved head. The genome's quality is completely unprecedented for an extinct species, providing the fundamental, high-resolution blueprint of the lost animal. However, having a digital genetic blueprint on a computer is only half the battle. To actually bring a living thylacine back, researchers must heavily edit the genome of a living surrogate species. Colossal identified the fat-tailed dunnart, a tiny, mouse-sized carnivorous marsupial, as the closest living relative and the ideal surrogate. Prior to this ambitious project, virtually no assisted reproductive technology existed for the dunnart. Driven completely by the demanding needs of the de-extinction project, Colossal's researchers successfully developed a reliable method to artificially induce ovulation in the fat-tailed dunnart, allowing them to collect multiple mature eggs simultaneously. Furthermore, they engineered a world-first artificial uterus device capable of successfully hosting marsupial embryos through the highly critical early stages of pregnancy. They have also managed to create the most highly edited animal cell line to date by successfully swapping over 300 distinct genetic edits into dunnart stem cells. The real-world implications of these dunnart breakthroughs stretch far, far beyond the resurrection of the thylacine. These exact same artificial reproductive and biobanking technologies are urgently and desperately needed for living, critically endangered species like the Kangaroo Island dunnart. Endemic only to the western end of Kangaroo Island, this tiny, fragile marsupial lost over 98% of its entire habitat in the catastrophic 2019 and 2020 bushfires, and the tiny surviving population of just a few hundred individuals is currently heavily preyed upon by feral cats. By utilizing the stem cell derivation, artificial ovulation, and complex embryo cultivation techniques pioneered by the de-extinction movement, conservationists now finally possess the tools to biobank the Kangaroo Island dunnart's unique genome and rapidly breed them in captivity, ensuring they do not follow the thylacine into the abyss of extinction. We have truly entered an era where conservation is no longer severely limited to merely fencing off fragmented habitats and just hoping for the best. The incredible convergence of traditional ecology, whole genome sequencing, assisted reproductive technologies, and cutting-edge synthetic biology has created a deeply powerful toolkit for genomic rescue. The highly successful genetic revitalization of the mountain pygmy possum definitively proved that targeted gene flow can banish the specter of inbreeding depression. The remarkable resilience of the Tasmanian devil's insurance populations confirms that we can successfully archive and protect wild genetic diversity. The monumental genomic studies on the koala reveal the hidden truth that populations can actively purge their genetic burdens through recombination if we just give them the demographic space to rapidly expand. And finally, the laboratory marvels of kangaroo IVF and dunnart reproductive engineering offer an unprecedented, sci-fi-level insurance policy against sudden environmental catastrophes. While the modern threats facing marsupials, from rampant climate change to devastating invasive predators, remain incredibly daunting, these monumental scientific breakthroughs completely rewrite the narrative of extinction. By actively and intelligently managing the genomic health of these species, we are restoring their evolutionary potential, fundamentally ensuring that Australia's unique marsupials continue to adapt, thrive, and define the continent's incredible ecological heritage for millennia to come.